Today we discuss megasporogenesis to fruits. First, megasporogenesis. Till last class, we discussed about development of ovule or type of ovule, or we discussed about the types of ovules. Now megasporogenesis. Megasporogenesis normally takes place in ovules. Megasporogenesis occurs in the ovules. Every ovule shows central diploid nucellus, which is nothing but megasporangium. It shows a funicle, chalaza, integuments, micropyle, and other things. During megasporogenesis, one of the cells in the hypodermal cells of nucellus converts into a special cell called archisporial cell. This chapter is important, that's why I am discussing. This archisporial cell is diploid, which divides and redivides to produce two cells. One is parietal cell, the other is sporogenous cell. Parietal cell divides and increases the amount of nucellus. Sporogenous cell divides and gives rise to megaspore mother cells. Sometimes archisporial cell directly give rise to megaspore mother cell. So, some of the hypodermal cells or one of the hypodermal cells in the nucellus transforms into archisporial cell. Archisporial cell divides and gives rise to parietal cell and sporogenous cell. Parietal cell increases the amount of nucellus. Sporogenous cell converts into megaspore mother cell. Now, nucellus has towards this end is micropyle end, towards this end is chalaza end. It has one megaspore mother cell. This megaspore mother cell, diploid, undergoes reduction division and produces 4 haploid megaspores. In these 4 megaspores, 3 degenerate, that means disintegrate. If 3 degenerate, only 1 remain functional towards the micropylar end. So, this is functional megaspore. This functional megaspore undergoes, now this is important for MSET. Now, this undergoes 3 times mitotic division. First time mitotic division, it produces 2 nuclei. Second time, out of 2, both are present at the micropylar end, one slowly comes to chalaza end. Now, one is at the micropylar end, one is at the chalaza end. Now, second time mitosis division takes place, 4 nuclei are formed, 2 at the micropylar end, 2 at the chalaza end. Third time mitotic division takes place, 8 nuclei are formed, 4 at the micropylar end and 4 at the chalaza end. Now, how many times mitotic divisions takes place means 1, 2, 3, 4, 5, 6 and 7 times mitotic divisions takes place. 3 times mitotic division, 7 mitotic division. The upper are called upper 4 polar nuclei, lower are called lower 4 polar nuclei. In this, one comes to the center and ultimately the arrangement will be 3 plus 2 plus 3. Now, till now, free nuclear divisions takes place without any cytoplasmic division. Now, cytoplasmic division takes place and cells are formed. This will form an 8-celled or 7-celled 8 nucleated embryo sac or female gametophyte. In this 7-celled 8 nucleated embryo sac, upper 3 cells are called egg apparatus with 2 synergids and 1 egg cell. The central 2 are called polar nuclei. The lower 3 are called antipodals. So, now if you take this structure, it appears like this. Gradually, the 2 polar nuclei fuses and ultimately a structure like this. 7-celled, 7 nucleated embryo sac is formed. These 2 are called synergids. This is called egg cell. This is called secondary nucleus formed by the fusion of polar nuclei. This is called antipodals. Now, in the synergids, these two, synergids and egg cell combinedly called egg apparatus. Now, these two synergids contain special finger-shaped structures called filiform apparatus. Filiform apparatus perform two functions. One is they collect the food from nucellus and supplies to the embryo sac, developing embryo sac, and it also secretes the chemical substances which guides the movement of pollen tube into the ovule. Egg cell involves in syngamy and secondary nucleus involves in triple fusion. Like this, embryo sac is formed, and this is called polygonum type of embryo sac because it was reported in Polygonum divaricatum by Strasburger. It is also called monosporic embryo sac because it develops from a single functional megaspore. This is the development of embryo sac. Next, pollination. The transfer of pollen grains from anther to the stigma is called pollination. Pollination in plants is of two types: direct pollination, indirect pollination. Direct pollination means pollen grains first reach the ovules because here ovary, style and stigma are absent. When pollen grains first reach the ovule, it is called direct pollination found in gymnosperms. Sometimes in some plants, pollen grains first reach the stigma, then goes to the ovule. It is called indirect pollination, which is found in angiosperms. In indirect pollination of two types: cleistogamy, pollination in a completely closed flower which never open in its lifetime. Chasmogamy, pollination in an opened flower. Chasmogamy is of two types: autogamy, pollination in a single flower; allogamy, pollination in between two flowers. Allogamy is again two types: geitonogamy, xenogamy. Geitonogamy means pollination between two flowers of the same plant. Xenogamy means pollination between two plants of the same species. Geitonogamy is pollination between two flowers of the same plant. It is almost a self-pollination. Xenogamy means pollination in between two flowers of different plant but belonging to the same species. This is introduction to pollination. Then comes the contrivances of cross-pollination. Contrivance of cross-pollination means for a plant, cross-pollination is more beneficial than self-pollination. Because by cross-pollination, disease-resistant characters are formed, more number of seeds are formed, seeds show maximum vigor and vitality, and there are so many advantages. So, plants try to perform self-pollination, try to avoid self-pollination and perform cross-pollination. The contrivance of cross-pollination are dicliny, dichogamy, hercogamy, heterostyly, pollen prepotency, self-sterility and sensitive stigmas. They are all contrivances. If any one of that is present, the plant perform cross-pollination and avoid self-pollination. Dicliny is presence of unisexual flowers. Dichogamy is maturation of essential organs, androecium and gynoecium at different times. Hercogamy means arrangement of essential organs in different heights or in different directions. Heterostyly means presence of different heights of styles in the flowers of the same plant. It may be diheterostyly or triheterostyly. Self-sterility means suppose the pollen grains reach the stigma of the same flower, they do not show any germination. Passiflora and Abutilon and orchids. Then pollen prepotency, domination of cross-pollination over self-pollination. Sensitive stigmas are very sensitive to the self-pollination, but they easily receive the pollen grains of other plants. Suppose cross-pollination fails, immediately plants perform self-pollination. For that they have some adaptations. The best example is Yucca gloriosa. The flowers of Yucca gloriosa shows symbiotic association with a special insect called Pronuba yuccacella. If that insect is there, the plant perform cross-pollination. If that insect is not there, the plant performs self-pollination. Homogamy, movement of floral parts, incomplete dichogamy, safety mechanism and cleistogamy are some of the things, but only three is given in our syllabus. One is homogamy. Presence of bisexual flowers and maturation of essential organs at the same time is homogamy, Caltha and Ranunculus. In these two plants, pollen grains are transferred from anther to stigma with the help of raindrops. Then safety mechanism. This is found only in the disc florets of Asteraceae. Here, the pollen grains, once the plant is protandrous, if cross-pollination fails, the stigmatic lobes bend downwards, touch the anthers and collects the pollen grains. This is called safety mechanism. And this safety mechanism is found in Asteraceae disc florets. Then cleistogamy. When a flower never open in its lifetime, there is no question of cross-pollination, there will be only self-pollination. Commelina bengalensis. Cleistogamous flowers are underground, chasmogamous flowers are aerial. Streptocarpus princeps, cleistogamous flowers are chasmogamous flowers, both are aerial. This is about the contrivances of self-pollination. Then agents of pollination. Pollen grains cannot move by themselves because they do not have any flagella or cilia or whatever locomotive structure. So they have to be taken from one place to other place by some agents, some external things. There are two types of agents. One is non-living agents, the other is living agents. Abiotic components, biotic components. Abiotic components are air and water. Biotic component is animal. Pollination by air is anemophily. Pollination by water is hydrophily. If pollination occurs on the surface of the water, it is epihydrophily, Vallisneria. Under the water, it is hypohydrophily, it is Zostera. Then pollination by animals, zoophily. Pollination by birds, ornithophily, Sterlitzia, Bignonia. Pollination by bats, Chiropterophily, Chygelia pinnata, sausage tree. Pollination by insects, entomophily, Cestrum nocturnum. Pollination by snails and slugs, Melacophily, which is found in Aroidae plants, Colocasia and other plants. Now, this is about the explanation of two lessons, megasporogenesis and pollination. Now, we discuss some important questions in this chapter. So, first question is, choose the correct sentence from the following in the development of angiospermic embryo sac. Choose the correct sequence means what will form first, what will form next, and what is the sequence of the formation. Megaspore mother cell, Archisporium, functional megaspore, embryo sac is wrong because first formed is Archisporium. Archisporium, megaspore mother cell, functional megaspore, embryo sac is correct because Archisporium divides and gives rise to megaspore mother cell. Megaspore mother cell divides by meiosis and produces 4 megaspores where 3 disintegrate, 1 remain functional. Then this megaspore undergoes numerous free nuclear divisions and later it produces cell wall resulting in the formation of embryo sac. So, the correct sequence from the following in the development of angiospermic embryo sac is Archisporium, megaspore mother cell, functional megaspore, embryo sac.